So it's really an enormous pleasure to introduce Lauren Rajakovich. So Lauren is a senior postdoc in my lab. She's actually also on the academic job market this fall. So Lauren did her undergraduate studies at Wake Forest University and then pursued PhD research at Penn State with Marty Bollinger and Karsten Krebs. So she really developed a deep expertise in understanding the mechanisms of metalloenzymes and joined my lab to pursue her interest in the discovery of new enzymes, with a particular focus on discovering enzymes from the gut microbiome. And so in her postdoc research, Lauren's been using her deep understanding of mechanistic enzymology to both identify gaps in our understanding of gut microbial metabolic activities, and also to discover the enzymes that are responsible for those activities. And so she's going to share a brand new story today. It's unpublished. And I'm excited about this story because I think it really illustrates how this microbial community is not only a source of enzymes, but also a source of enzymes with unique chemistry.
Thanks, everyone. Thanks, Emily, for the introduction. Like she said, I'm excited to share with you as an enzymologist why I got interested in the field of the human gut microbiota. And it's really one of the most exciting things is this untapped potential to discover new chemistry and enzymes in these types of bacteria. So a very recent study looked at the proteins encoded in gut microbes and compared them to just generic protein databases, and found that about 40% of proteins encoded by gut microbes cannot be matched to any proteins that we have an assigned function for in these protein databases. 27% don't match to any protein at all. 14% do match to a protein in these databases, but the function is totally unknown. And then the remaining 60% match to annotated proteins, but as I'm sure many of you in the audience are aware, automated annotation of proteins can often be inaccurate or not specific enough to assign a function to a protein. Now, the chemistry that we know about in the human gut microbiota is really exciting. Microbes can perform challenging chemistry such as dehydroxylations and decarboxylations in the absence of oxygen. And these transformations can have really important consequences for host biology. Metabolism of bile acids generates a whole class of signaling molecules involved in metabolic responses. Catechol dehydroxylation can alter levels of neurotransmitters potentially, and also metabolize dietary nutrients that have this motif. And aromatic decarboxylation from amino acid metabolism can generate disease-associated molecules like paracresol. Now, in the Balskis lab, we're not only interested in discovering new chemistry, but like Emily said, in understanding the enzymatic transformations that enable this type of metabolism. And our hope is that by uncovering these enzymes and understanding this chemistry, we can have a molecular mechanism, mechanistic understanding of how the microbiota can influence the human host and its disease or health states. And this can help us to elucidate correlative versus causative relationships and also enable potential therapeutic translation of these molecular mechanisms. And so one of the types of chemistry that we're really interested in the Balskis lab is the transformation of dietary nutrients that have this common quaternary amine motif and its metabolism by gut microbes into the small molecule trimethylamine. Now, trimethylamine is a uniquely microbial metabolite, but is reabsorbed by the host and oxidized in the liver into trimethylamine N-oxide, or TMAO. And the reason that we're interested in this type of chemistry from a biological standpoint is that TMAO has been associated with increased risk for a number of different human diseases. And so my project has been focused on how carnitine is transformed into trimethylamine by gut microbes. Carnitine is an essential host nutrient consumed in high quantities daily and is really abundant in red meat. And it's important for the host to ingest this nutrient because it's essential for fatty acid metabolism. It acts as a carrier for fatty acids into the across the mitochondrial membrane. But like I alluded to on the previous slide, this metabolism by gut microbes of carnitine into TMA has been associated in particular with cardiovascular disease. And so we wanted to understand how this transformation is occurring. Now, a few years ago, a pathway was discovered in certain facultative aerobic bacteria where a risky type monooxygenase called CNTA hydroxylates carnitine at the C4 position, followed by non-enzymatic degradation of this molecule to generate trimethylamine. However, the sort of relevance of this metabolism in the context of the human gut has come into question because this requires molecular oxygen for the reactivity. And in the human gut, it is largely anoxic, especially in the colon. Other studies looking at bioinformatically, the abundance of this CNTA gene in metagenomes have found that it's not correlated with plasma levels of TMAO. And in addition, looking at metatranscriptomic data, that although the CNTA gene is present in metagenomes, no transcripts have been detected in these two data sets that were analyzed. And so this got us thinking about other ways that carnitine could be metabolized to trimethylamine in the gut. Now, there was some evidence of an anaerobic pathway, actually dating all the way back to the 80s, where carnitine is transformed into gamma-butyrobating under anaerobic respiration conditions of some bacteria using the Chi genes. However, this second transformation was really not well established. In my own work that I won't have time to talk about, I looked at metabolism of carnitine and gamma-butyrobating in complex communities of bacteria cultured from human fecal samples, and was able to show that actually this anaerobic pathway is occurring in these communities, but it was unclear what types of bacteria were performing this second reaction. In fact, there's only one single bacteria that's been reported to do this transformation under anaerobic conditions. It's called Emergentia timonensis, but the molecular mechanism of this transformation was unknown. So that really became the focus of my project, is understanding how this chemistry is happening. As you can probably tell, this is a really challenging enzymatic transformation to catalyze, to cleave this carbon-nitrogen bond here. And so we wanted to then uncover the enzymes in chemistry occurring here and use that information to answer questions about how distributed this chemistry is in other microbes. And importantly, what is the relevance of this metabolism in the human body? And so again, just to start with thinking about how this carbon-nitrogen bond could potentially be cleaved, I looked into the literature to see if there were other known enzymes that I could apply to this system. And there are actually only 3 enzymes that are known to generate trimethylamine by cleaving this carbon-nitrogen bond. They're choline TMA lyase, glycine betaine reductase, and ergothionease. And as you might be able to tell just by looking at these substrates, they differ from gamma-butyrobating in the fact that each of them has a sort of functional group attached to the carbon adjacent to the CN bond to be cleaved. And this could potentially activate these molecules for carbon-nitrogen bond cleavage. Gamma-butyrobating lacks these functional groups nearby. Just thinking about more mechanistically what we know about these enzymes, choline TMA lyase is a glycyl radical enzyme that uses protein-based radicals to generate this key ketyl radical anion intermediate that leads to the cleavage of the carbon-nitrogen bond in this enzyme reaction. Now, glycine betaine reductase, as the name implies, catalyzes a 2-electron reduction that uses a selenocysteine protein residue to attack the alpha carbon of the substrate, releasing trimethylamine and forming a selenoether adduct on the protein backbone. Now, the last example, ergothionease, is also a redox neutral transformation like choline TMA lyase, but this mechanism is unknown. However, it's possible that this sort of imidazole-like functional group could enable a sort of Hofmann-type elimination that you might be familiar with from organic chemistry, where the proton of the carbon adjacent is abstracted, resulting in C-end bond cleavage. However, the proton here in the Hofmann elimination needs to be have a pKa that's sufficiently low enough under biological conditions for this reaction to occur, and that's potentially how this imidazole could participate. However, in the case of gamma-butyrobating, the pKa of this proton is approximately 40, which is much too high to enable this type of chemistry without some enzymatic help. And so what I felt like I needed to know was sort of what is the overall chemical transformation occurring here in order to have some more insight into the enzyme chemistry. And so I set out to determine what the co-product of the reaction is. And so in cultures of Emergentia timonensis incubated with GBB, I looked for production of carboxylate containing products and found that butyrate was being generated on a similar timescale to trimethylamine production in these incubations. I used isotopically labeled substrate to confirm the origin of this butyrate from gamma-butyrobatine or GBB. But when I looked at the mass spectrum of the butyrate being produced, what I was looking for was the sort of retention of all 6 protons, all 6 deuteria from the labeled substrate in the case of a 2-electron reduction. However, this is not what I observed. Instead, I saw the major isotope detected have only 4 deuteria remaining. And what this suggested to me was that perhaps there was an intermediate being generated before butyrate that had an unsaturation, such as this intermediate crotonate, that would result in loss of 2 deuteria in the final butyrate product. And this is a really key conclusion to make because it gave me a real clue as to how GBB might be metabolized. I mentioned I looked in the literature for TMA generating enzymes, but I also looked to see how other molecules that look like GBB might be metabolized. And what I came across was this pathway in gut microbes actually for metabolism of GABA, where it's first transformed into 4-hydroxybutyryl-CoA. And then there's a C4 elimination of water to generate crotinyl-CoA and then finally reduced to butyryl-CoA. And so using this as a template, I came up with the hypothesis for how GBB was metabolized, first forming this CoA thioester, and then a C4 elimination of a better leaving group in the trimethylamine, and then following downstream reactions in a similar fashion. I looked for these putative intermediates using isotopically labeled substrate again in Emergentia timonensis, and was able to observe accumulation of this CoA thioester of GBB, as well as deuterium incorporation into the final butyryl-CoA product here. However, I was still left with the question, what are the enzymes responsible for this chemistry? And so I used an untargeted approach to look at the transcriptional response of Emergentia timonensis to GBB. And the reason I thought this would work was because of a previous experiment that I had done where I grew Emergentia timonensis in the presence or absence of GBB, then spun down the cells, resuspended them in buffer, and incubated them again with GBB, and only saw conversion to trimethylamine when the cells had been pre-grown with GBB in the media condition. So this suggested that the presence of GBB was inducing the metabolic machinery necessary to generate TMA. So looking in an RNA sequencing experiment, I looked for genes that were both highly expressed as well as differentially expressed in these 2 growth conditions. And so this upper right quadrant identifies 6 genes that could be potential metabolic enzymes. And these 6 genes are co-localized in the genome of Emergentia timonensis, and this is pretty common for metabolic pathways in bacteria. And so I attempted to connect these genes and their putative annotations with the hypothesis for metabolism that I had generated. And so we sort of knew we were maybe on the right track by the presence of this transporter gene that encodes a protein that's specific for molecules with quaternary amines. Then the next step of forming the CoA thioester could be catalyzed by the 2 CoA transferases encoded in this gene cluster. And actually what I found was that taking these proteins in vitro, you do generate GBB-CoA from GBB. However, this only happens when you have both proteins together in the assay. Neither one of them on their own can do this reaction, suggesting some sort of a protein complex. And also found that the preferred substrate for as the CoA donor is acetyl-CoA. The next and of course most interesting reaction is this carbon-nitrogen bond cleavage. And as you might suspect, none of those enzymes I brought up earlier in the talk were found in this gene cluster, that it's totally new type of chemistry. And so I hypothesized that maybe BBUA, this Acyl-CoA dehydrogenase-like protein, could be involved, and I'll explain a bit more why in the next couple of slides. But in fact, including BBUA and heterologous expression of these first three genes in E. coli led to production of TMA from GBB. And then I confirmed this in vitro reactions with these three proteins, demonstrating both multiple turnovers to generate trimethylamine, as well as production of crotinyl-CoA, the proposed co-product of this reaction. Just a little bit of background, as I'm sure many of you know, Acyl-CoA dehydrogenases typically are flavin-dependent enzymes that do 2-electron redox chemistry, involving hydride transfer to install an alpha-beta saturation or to remove one. However, the reaction I'm talking about here is actually, a redox neutral transformation to generate TMA. And what I didn't mention before is in the GABA pathway that this 4-hydroxybutyryl-CoA dehydratase is also a flavin-dependent enzyme of this Acyl-CoA dehydrogenase-like family. However, these two proteins share less than 10% sequence identity to one another. So despite catalyzing potentially a very similar reaction, they don't seem to be close homologs to one another. And so I used the information of what we know about how Acyl-CoA dehydrogenases use their flavin cofactor, either in 2-electron chemistry or via 2 single 1-electron transfers, as well as some mechanistic hypotheses for the dehydration reaction to come up with this hypothesis for how GBB is transformed into crotinyl-CoA and TMA. So as I mentioned earlier, the challenge of this reaction is sort of the unactivation of these carbon-hydrogen bonds in GBB. So the first solution was to generate the CoA thioester, which reduces the pKa of the alpha proton, which enables formation of the enolate. Now, from this stage, the flavin cofactor could act as a 1-electron oxidant to generate a ketyl radical. And now this species has a much reduced pKa of this C3 proton. And the reason that this pKa is so reduced is because the resultant product of proton abstraction it has a number of different resonance forms. And the most important resonance form is this ketyl radical anion that I'm showing here that could result in elimination of a C4 functional group to generate TMA and a ketyldiene product. Now, this should maybe remind you of the choline TMA lyase mechanism that uses a ketyl radical anion for carbon-nitrogen bond cleavage. So potentially similar mechanistic logic with a totally different type of enzyme. And then the next step would be 1-electron reduction of the substrate, giving you the overall redox neutral transformation and formation of the crotinyl-CoA product. Now, this at this point is just a proposed mechanism. I'm really excited to investigate these proposed steps through more detailed experiments in the coming weeks, and really more than weeks. And so just to take you back to sort of our framework here, we're able to understand now the chemistry and enzymes that enable this transformation. And I just want to give you, in the last few minutes, a little taste of how we can use this information in the context of the gut microbiota to learn more about biology. So what I did was take these BBU genes and look for them in other microbes, since Emergensia timonensis was the only one known to perform this chemistry previously. And what I found was only a handful of bacteria that have a similar looking gene cluster in their genomes. And these are restricted to host bacteria that are obligate anaerobes of the Clostridiales order. And a large majority of them are actually uncultivated bacteria. Their genomes were computationally assembled. And so this sort of highlights, as I brought up in the beginning, the potential to discover new types of enzymes and chemistry in the vast genetic space of uncultivated bacteria. then what I did was to take these genes and look in human samples, looking at metagenomes metagenomes and metabolomes, where I looked to see if the presence of these BBU genes could be correlated with metabolite levels or disease of human populations. And what I found by looking at the metabolomics data associated was that when BBUA was present in the metagenomes, you have lower levels of GBB in the stool. And then looking at cardiovascular disease cohorts, found an increased abundance of the BBU genes in the cardiovascular disease cohort compared to healthy controls. So again, just back to our roadmap, hopefully what I've been able to show you today is how we can understand new chemistry in the gut microbiota, discovering a new carbon-nitrogen bond cleaving enzyme that uses a flavin cofactor to release trimethylamine. We can then use our knowledge of these genes and enzymes to find this metabolism in other bacteria. And one thing that I want to note is that bacteria that have this chemistry do not have this first type of chemistry, requiring sort of cooperation and co-metabolism in the gut microbiota to make trimethylamine. And then finally, we can use metagenomics and metabolomics to evaluate whether this metabolism is actually relevant in human samples, looking at changes in metabolite levels, and then potentially looking at disease cohorts. So with that, I'd like to thank the Balskis Lab for all of their support, in particular Emily on this project. Maud Bullenbach synthesized the isotopically labeled substrates. Beverly Fu is a graduate student who really helped me with the bioinformatics pipeline. The Broad Microbial Omics Core for the RNA sequencing, and finally funding sources, and I'll take any questions. Thank you.
Thanks so much, Lauren. Great talk. We're a little tight on time, so we might have time for one question, and any unanswered questions you can answer in the Q&A function.
Hey, Lauren, fantastic talk. So the first question comes from Wolfgang Krutel, who indicates, great talk, Lauren. Could you imagine that the reaction could run in the reverse direction?
Yes, I think that that's an interesting way to probe the mechanism that I'm proposing. You could actually imagine that, sorry, I'll go back to the mechanism really quickly, that you can look at the isomerization reaction on its own, and then you could also think about running the reaction with TMA to generate GBB-CoA. So I think that's definitely possible, and is something that has been investigated with the dehydratase enzyme that I was comparing it to.
Okay, there's kind of quite a few questions flooding in. Andrew, do you have time for one more, or do you want to move on?
Let's do a couple more questions. This should be fine.
Okay, cool. So Lauren, could you go over the substrate specificity of these enzymes? Or, I mean, just a quick question, can they act on other substrates?
Yes, so I have tested the CoA transferases with carnitine and with GABA, so structurally similar molecules, and they do not act on those substrates. So it seems like that could be potentially the first degree of specificity in this pathway. I will admit that I recently was only able to isolate the BBUA protein to start studying it in vitro recently. It really did not want to express solubly. I had to co-express the chaperones from Emergentia tuminensis. So I haven't actually gotten to look at the substrate scope of this enzyme in particular, but that would be exciting to do.
Okay, and then there's a couple of questions on plausible mechanistic alternatives to what you proposed. So I'll just very quickly go with the first one from Stephen Wallace, and then maybe we'll take the one from Wilfred van der Donk offline, maybe. So Stephen asked if you've anticipated an N-terminal oxidation followed by COPE type eliminations.
So I haven't thought about it. I guess the challenge with doing the oxidation, so if I'm understanding correctly, the two electron oxidation to generate the alpha beta unsaturation, then you would need to attack the least electrophilic carbon to have that elimination occur. But I guess there are plenty of mechanistic possibilities to explore, and that will be really interesting to identify.